Good afternoon, everybody. So yes, I'm from MatéIS lab at INSA Lyon, and yes, at the microscopy team, we are working on some new techniques of microscopy. The goal is to go deeper and faster to understand better the materials. So I'm gonna present you our work. It's at the border of many fields, 3D reconstruction of hydrated samples at the sub-micrometric scale. Usually, when we do microscopy, it's to study dry samples because the high vacuum chamber of a microscope is not very compatible with liquid state. But there are lots of hydrated materials worth to be studied and some processes, and it's why for years environmental microscopes are developed. They are specific microscopes allowing gas to be in the chamber and to study materials in hydrated state. The easy sample preparation with achieving high resolution, it allows to do some in situ studies, which is perfect for material science and biology. One drawback is a beam effect, which can damage our sample. In this context, we have five challenges to overcome at the same time. The first one is liquid electron microscopy. It's not so easy to have a stable sample in hydrated state, even in environmental microscope. Second one is 3D reconstruction, how to go from 2D images to 3D model, which can be analyzed. And the third one is sensitive materials. There are a lot of material, especially biologic ones, which are very sensitive to electron beam. Spatial resolution, the maximum theoretical resolution in a microscope, like in an ISM, is a few nanometers, and we are trying to get close. And the last one is time resolution. Without speaking yet of dynamical studies, it's very important to be fast enough to acquire an entire 3D dataset before our sample can move, change, or be damaged. Our answer for that is the technique of STEAM in eSIM. It's basically doing scanning transmission electron microscopy and tomography in an environmental scanning electron microscope. So I said it's not so easy to maintain our sample in a liquid state, but it's possible because in an environmental SEM and with a dedicated sample holder, we can precisely tune both the pressure and the temperature in the chamber, and so tune precisely the relative humidity of our sample, and we can set it to control the water layer thickness or droplet size, and using our sample holder in transmission mode, putting our detector just below our sample, it's possible not only to have the secondary electrons or back-scattered electrons, but also analysis in bright field, dark field, or high-angle annular dark field mode. So it's a lot of information we can have at the same time. The resolution here is limited by the chamber pressure and also by the water layer thickness. So if it's under 1 micrometer, we can have 10 nanometers and more of resolutions. And now we have our 2D images of liquid state samples, and how to go to the 3D models. It's called the tilt series acquisition. It's like a medical scanner, but here we are tilting our sample step by step, angle by angle, and taking images And by inverse Radon transform, it can give us quantitative model which can be analyzed after. There are main limitations, but it's a technique which works very well. So here there is our dedicated stage. It's a lab-made stage. There is some piezo controller to set our eccentric position. There is a Peltier stage to tune the temperature, and it allows us to control both the position, the temperature, and the pressure in the microscope. So it's possible to go from these 2D cell images. It's a mouse fibroblast fixed but hydrated cell to this model. So to this 3D model. So it's an entire whole cell in his liquid state, in its liquid state, and yes, it's a recent work we succeeded in some months ago. And our last drawback we are working on is irradiation damage, because when an electron hits water or an organic sample, it's like a firework of a chemical species. There are a lot of reactions, especially the formation of free radicals, and it can damage our sample very quickly. So we have some solutions, essentially concerning time control. We have to do our experiments very fast. For example, you do not set all your parameters on your samples. You are doing it just close the sample on the axis of rotation. You reduce exposure time by image, doing just what you need, taking the lowest number of imaging you can, and taking the minimum time between the start and the end of your acquisition. And it's for that we are doing a lot of work for automation, allowing us a very precise time and dose control, yes, to have very precise parameters. So for my conclusions and some perspectives, liquid phase STEAM allows the study of hydrated materials at the same time in secondary electron, bright field, and HAADF modes. Acquisition of tilt series is possible in liquid phase STEAM to produce 3D models around 10 nanometers resolution. Whole and hydrated sample cells can be analyzed with 3D liquid phase STEAM. And for the future, experiments will be due to optimize electron dose and control water layer thickness very well, and further investigations for images post-processing and tomograms generation, which is a huge part coming after this acquisition. And yes, we are working on that. Thank you for your attention.